Thank you very much, Fred, for the kind introduction, and I'm really honored to be here today and receive this award. I came into lipid research, and I do agree with Fred, without Avanti lipids, I think none of the stuff that you will see today will be possible to be done. So I will start with just an introduction about the lab. We are actually studying mostly lysosomal biology and biochemistry, and it turns out that one of the major aspects of lysosome biology and biochemistry is degrading lipids and dealing with lipid recycling. And if you think where this would be most important, it turns out it's in the brain. And this is why my journey ended up studying lysosomal lipid catabolism in brain diseases, especially neurodegeneration. And I will show you some stories that came from the lab just focusing about this aspect of lysosome biology. So just to be on the same page, the way we look at the lysosome is like any other biologist studying this organelle. This is the place where almost all types of biomolecules get degraded. And the degradation of these molecules is guided by the flux of material to the lysosome, either through endocytosis or through autophagy, where macromolecules are being delivered to the lysosome to be recycled, and their content being released again to the cytosol. But also lysosome is important QC component of the cellular system. It degrades faulty organelle, again recycling their content. The importance of the lysosome is really showing up by looking at the genetics of human disease. It turns out that we have almost 50 to 60 different lysosomal storage diseases. We recently discovered a new one. These were heavily studied in the past, where we have mutations directly in lysosomal genes that encode lysosomal proteins, either hydrolases or transporters. So in this way, we have material stuck in the lysosome that doesn't get out, leading to what we call lysosomal storage diseases. 70% of those are affecting the brain, and that's why we think lysosome is really important in the brain. However, really the revolution and the understanding of lysosome biology came into play more recently when we started to realize that age-associated diseases, these are complex but much more common than the ultra-rare lysosomal storage diseases. It turns out the genetics behind those diseases refer most of the time to the lysosome, especially in the case of Parkinson's disease. So if you take US studies or familial forms, in many cases, these are the same lysosomal genes that cause lysosomal storage diseases, but in this case, only one allele is mutated. The other one is spared. Most probably that's why it takes some time until these people suffer from neurodegeneration. So my lab is focused on studying monogenic diseases to understand these complex ones. And the question that we ask is so simple. What is exactly happening to the lysosome when we have these mutations? How can we define at the biochemical level the lysosomal dysfunction? And the way we do this is by directly looking at the small molecules inside the lysosome. So if the function of the lysosome is to degrade macromolecules into small ones, the best way to analyze its functionality is to have quantitative tools that allow you to measure those degradation products. We do so with a method that Fred mentioned, which is the lyso-IP that I developed when I was a postdoc in the Sabatini lab, where we tag the lysosome with a membrane protein that has an epitope. Now you can take the cells, break them open, release the organelles, come with magnetic beads that would allow you to pull down these lysosomes, and you get very enriched and sometimes ultra-pure lysosomal prep, depending on the cell type. With these lysosomes, you can do anything you want, from biochemistry to quantitative omics analysis, and you can even do patch clamping and other things. So the thing here now, you have a very simple platform where you can engineer these mutations in human diseases in cell culture, and then you can purify lysosomes and look exactly what's happening, and not only looking at the whole cell changes that usually people used to study in the past. When we decided to apply this technique and started my lab, I thought that I should be focusing on interesting diseases where the gene function behind those diseases is not understood yet. And we focused on this group of disease called NCLs, neuronal ceroid-lipofuscinosis. This is a neurodegenerative disease in kids where kids have dementia or Parkinsonism phenotypes similar to the aged individuals, but the age of 5 to the age of 10. So very severe neurodegenerative diseases. In kids, collectively, they are the most common neurodegeneration in kids. But they are still rare diseases. They are caused by mutations in 14 different genes. These genes have different functions, and in most cases, we don't know the exact function of these genes, but they are related to the lysosome. When we started, we focused on CLN3 that causes the most common form. It's also called Batten disease. And the disease, as I mentioned, is very severe. By the age of 5, the kids will have vision failure, followed by typical neurodegenerative disease with mental decline, seizures, motor skills dysfunction, and very early death. So CLN3 function was not known back then, but also we did not really know anything about the disease molecular pathology, and we only knew that it's a membrane protein on the lysosome. We said, let's take this one, knock out the CLN3 in cells, pull down the lysosomes, and see what's changing. And it turns out when you knock out CLN3, you have this massive accumulation of the glycerophosphodieesters of the phospholipids. So you cleave the two acyl groups, you end up with the glycerophosphodieesters, or we call them GPDs here, that accumulate massively inside the lysosome. If you profile the cells or the brains, you see nothing. You only see it when you really look inside the lysosome. So this led us to hypothesize that there is what we call extra-lysosomal nutrient supply deficiency, because these GPDs do have important neuronal nutrients, including choline, inositol, and serine, and we showed this in the paper. But what's interesting, we now have also potential biomarker for this disease. So we developed a tagless lyso IP with our collaborator that allow us to pull down lysosomes from patient samples, and we did see exactly the same thing. And physicians now are using this approach to define if variants in CLN3 are actually pathological or not, which is very rewarding starting from a basic science work here. But we were also busy trying to know what exactly happens to the lysosomes once you accumulate those GPDs, why they are bad. It turns out that these glycerophosphodieesters inhibit specifically the second step of phospholipid degradation, causing massive accumulation of the lysophospholipid, which is the intermediate, inside the lysosome. And this is a disruptive lipid because from a biophysical point of view, it's very bad for the membranes to have large amounts of these lysophospholipids inside them. But when we were working on this biochemistry and using kinetics, we realized that the first step on degrading lipids is 1,200 times faster than the second one. So if you think about it from a very simple mathematics, you would realize that you have 1,200-fold more of this lysophospholipid accumulating. And that doesn't make sense because I just told you these lipids are so bad for the lysosome. So Sam and Wentao discovered that there is a whole new pathway in the lysosome that allow these lysolipids to get out of the lysosome for a quick recycling by reacylation through the Lance cycle, which is really interesting to us because when we found this gene, we said there must be a neurodegenerative disease because that fits with the idea that lysolipids are bad, but no one reported any disease like this back then. But a year after, there was already patients collected showing that we have an autosomal recessive neurodegenerative disease in humans with Spencer Spencer null mutations. And if you knock out Spencer in the CNS, you get, again, a typical neurodegenerative disease phenotype. So this all shows you that lipid catabolism, phospholipid catabolism in the lysosome is really tightly linked to neurodegeneration, something we did not appreciate before all this work because in the past we always focused on sphingolipids and glycosphingolipids, especially in the cases like Gaucher disease and gangliosidosis and other diseases where we know that the culprit are enzymes that degrade them. So Uche Meadow, who used to be a grad student in the lab and now has his own lab, decided to do the same approach with this protein CLN5, so another causal gene for for Batten disease. And this is now a luminal protein. It's inside the lysosome, but causes the same disease as CLN3. And what he did is a very simple experiment. Again, take HEC293Ts, knock out CLN5, take the lysosome, throw them on mass spec, and see what's going on there. And he got this really beautiful volcano plot where you have accumulation of lysophosphatidylglycerol, depletion of a lipid we call BMP, or bis-monoacyl glycerophosphate. And bis-monoacyl glycerophosphate is an isomer of PG, phosphatidylglycerol. Being in the lysosome space, this is really super interesting because BMP is actually a strictly endolysosomal lipid. It's in the endolysosomal system. And what's even more important, we did not know how BMP is made or what is the machinery that makes BMP for many, many years. But just to tell you why BMP is very important for lysosome biology, it turns out that inside the lysosome, you need to degrade, as I mentioned, lots of these lipids that come in, including glycolipids and others. And to do so, you need these intra-lysosomal vesicles that I'm simplifying here. To create them, you need stable lipid that create them, and this is the BMP. BMP is an ultra-stable lipid inside the lysosomal environments. It's not being degraded by the hydrolases, but it also functions based on a lot of work done by pioneering scientists in the 90s and the 2000s, showing that this intralysosomal vesicle platform is a docking place for all the different lipid-degrading enzymes in the lysosome. This includes GBA, the glucocerebrosidase, ASA, the acid ceramidase, and many others. They dock there, they get close to their lipid substrates, and then they degrade them. And these are the same risk factors for many neurodegenerative diseases. So we believe that if you actually increase BMP levels, you might be able to reactivate them, and then you can ameliorate some of these neurodegenerative diseases. And that's why people were interested in knowing what is the BMP synthase, so you can boost BMP synthesis. Another major aspect of BMP biology is really the cholesterol efflux from the lysosome. Work from Grunberg and Judith Storch showed really nicely that we have an independent pathway of the NPC1 that depends on NPC2, Niemann-Pick type C, that allows the cholesterol to be effluxed and from lysosome, and this is also dependent on the BMP ladin, on BMP-containing vesicles. So you can imagine how important BMP to the system, and you can imagine that an enzyme that makes BMP would cause neurodegeneration if it fails to make BMP. So back then, we thought this is the pathway, like all people in this field. The BMP is made from two LPG molecules. Usually these will be generated most probably from the cleavage of acyl chain from the phosphatidylglycerol. And then this reaction is happening in the lysosome, the LPG to BMP transformation. So what we saw is decrease in BMP, increase in LPG. You don't need to be a rocket scientist to propose maybe CLN5 is a factor for this synthesis or the enzyme itself. Using very rigorous biochemistry, it turns out that indeed CLN5 is the BMP synthase that have been long sought since the discovery of the BMP itself, where through a very simple deacylation transacylation reaction, you can make the BMP molecule. When we published this, we did not have in vivo models. We just did this in cells as well as in iPSC-derived neurons. But now we have the mouse, and as you can see, when you knock out CLN5, you almost completely lose the BMPs from any tissue you test. This is log 10 fold changes. Really, the remaining amount is around the noise. We also got access to the sheep model, which was used historically to develop the gene therapy for these diseases. And we see exactly the same thing. And we also showed that if you do gene therapy, you have a correlation between the success of the gene therapy approach and the BMP recovery. But what most important for us was to show that the lysosomes are no longer, the in vivo ones, are no longer making BMP if you lose CLN5. you can do this experiment really easily with our tools. You pull down lysosomes from the liver within a very short period. Then you mix the lysate with the D5-LPG precursor. You measure BMP synthesis then. And this is what you get. If you have a knockout of CLN5 in red, you have complete depletion or inability of the lysate to make any BMP anymore. You can rescue this by throwing some CLN5 in the lysate. Our most exciting data is really this data. Again, like CLN3, we wanted to go to patients, and in patients of CLN5, we clearly see depletion of BMP in the variants that are available to us. They are not completely null. They still have some activity, but we can see this in the dried blood spot as well as in the plasma, and this provides now a very nice interventional biomarker for the gene therapy study that is ongoing currently by Neurogene. So this is really the part about understanding the disease is exciting, but the more exciting part to us is the fact that now we have a handle of this BMP pathway, because we know that enhancing BMP synthesis in the lysosome by throwing BMPs on the cells, let them take the BMP and going to the lysosome, we already know that we can enhance lipid catabolism. Now we have a target, a drug target, which is the CLN5 enzyme itself, and we think that this can be a candidate or a target to probably treat several different neurodegenerative diseases where the dysfunction depends on a dysfunctionality in some aspect of lipid catabolism in the lysosome. But if you are in the drug hunting space, you realize that it's really super hard to activate enzymes, much easier to inhibit them. And this is why we were really interested in understanding if there is any turnover of this ultra-stable lipid. And Kwamina and Gian in the lab realized that this is the case by really doing these biochemical fractionation studies. And here I'm just summarizing their data, their results. So if CLN5 is making BMP, it turns out that PLA2G15 they discovered is hydrolyzing this BMP. You can see this nicely here if you use an SS, the specific stereochemistry of BMP that is in the endogenous system. You can take this BMP, incubate with PLA2G15. You see very nice degradation of this lipid. But BMP is still ultra-stable. How does it maintain its stability inside the lysosome? It turns out that the BMP that we make is actually this BMP, which is an SS stereochemistry on the two chiral centers, but very important to the stability is the acyl position. The one you buy from Avanti is actually one where the primary alcohol is acylated, but the one that we have in the endogenous system is the one that have the acyl chain on the secondary carbon. So it turns out the combination of the stereochemistry as well as the regiochemistry is what keeps it stable. PLA2G15 cannot touch it even if you incubate it overnight. But there is a process of acyl migration that we still don't understand that produces this vulnerable BMP that is still the same stereochemistry but different regiochemistry, and this is the one PLA can degrade. The question if this is physiological, it turns out to be yes, it is. If you knock out PLA2G15 in tissues, in cells, you see very nice increase across tissues of these BMP species. So we think this is one way to boost BMP levels. We were so excited, we decided let's now test the therapeutic hypothesis of BMP. You can simply choose any of these models that I showed before. We chose the Niemann-Pick type C1 because this model is well established by work by Judith Storch, where she used to treat the cells with BMP directly, or PG, the precursor. So in this case, NPC1 is mutated or knocked out, and then cholesterol accumulate in the lysosome. But you can bypass this by inducing NPC2 clearance of cholesterol. We don't know exactly how this is happening. So we knocked down in patient cells. We knocked down PLA2G15, and we indeed saw very nice decrease using two different siRNAs here in two different patient cell lines of the cholesterol levels. But we got lucky because it turns out a company actually got to the same conclusion that PLA is a modifier in NPC, but they did not really know how it does it until we showed them our work on BMPs. They already crossed PLA2G15 mouse directly with the NPC1 knockout model, the most severe of all the Niemann-Pick type C models. And this is what you get: Purkinje cells, almost completely gone in the NPC. This is a hallmark of this disease. You cross them with PLA2G15, you actually preserve the Purkinje cells from death, which is a very, very striking result. And what's important is that we can move the needle on the survival by almost 65% in this very severe model, as I mentioned, where there is a complete loss of Niemann-Pick type C1. So this is the death within 70 days, a median of 70 days survival. And you can see that PLA2G15 double knockout with the NPC would make them survive much more. So we are really excited about these results, and that's why we are still now looking at other regulators of the BMP pathway. We do have now the enzymes that make or grant that very specific stereochemistry of the BMP. We know how the precursor is being channeled from the ER, where it's originally made, into the lysosome. So different components of the pathway are being elucidated now by us and also by other people in the field. And we think each one of those can be a drug target, and then we can hopefully boost the BMP levels and try to see if we can ameliorate different types of diseases, something we are also doing in the lab to provide proof of principle of the therapeutic potential of the pathway. With this, I would like to thank my amazing team, like really brilliant students and postdocs and research associates, as well as my collaborators, both on the clinical and the basic science side, as well as, of course, our funding. And thank you very much for listening.